So, in the last video, we studied about two techniques, that is crowded plate technique and oxanographic technique of primary screening. So, this time we are going to study the third method of primary screening, that is called as enrichment culture technique. Now, this enrichment culture technique is especially utilized for isolating desired microorganisms from a huge heterogeneous microbial population. Say for example, if we want to isolate enzyme producing organism from soil. Okay, the technique of enrichment culture, the basically here the medium is enriched, the medium that is utilized for isolating microorganism is enriched with specific substances which will support the growth of only particular kind of organism that is desirable to us. It will not support the growth of other unwanted microorganism, and that is how we can isolate a particular typical microorganism desired by us from a heterogeneous microbial population. Okay, so for that what we do is nutrient broth or nutrient medium is inoculated with microbial source like soil. A small portion of the inoculum is plated on solid medium and well isolated colonies are obtained, and the suspected colonies from the plate are further subcultured. Okay, let us take an example of, say, if we want to isolate enzyme producing organism from soil. Say, which enzyme? If we want to isolate amylase enzyme producing organism from soil. If you take 1 gram of soil, it will contain millions and millions of organism, but amylase producing organism will be very few. So we want to isolate that organism which is present in less number in the microbial population. Say amylase enzyme producing organism from the soil. So first thing we do is we prepare serial dilution of the soil sample, sterilize the diluted sample by heating to 80 degree so that it will kill all the vegetative cells. Only spores will survive. Okay. Then we use a enrichment medium, a medium which contain a particular ingredient that will support the growth of amylase enzyme producing organism. So for that we use medium containing starch. So we use starch agar. Plating is done on starch agar, and after inoculation the plates are incubated, and after incubation we have to identify which are the colonies that are formed by amylase producing organism. So what we do is after inoculation and incubation we pour iodine into the plate. Okay, and check the colonies that are surrounded by clear zone of hydrolysis. See, the medium contains starch. When you add iodine, and starch react with each other and a blue-violet color is formed. Okay, so starch agar will turn blue-violet if starch is present in the medium. Now those organisms, those colonies of microorganisms which can produce enzyme amylase will degrade starch, will hydrolyze starch and convert it into smaller products. When the starch surrounding a colony is hydrolyzed, it will not react with iodine and it will not show any blue color. So, in the plate, after incubation, we add iodine and check those colonies which are surrounded by a colorless clear zone of starch hydrolysis. Those colonies which are surrounded by a colorless clear zone of starch hydrolysis are produced by microorganisms which are amylase enzyme producers. Such colonies surrounded by clear zone are picked up, further subcultured and purified and used for amylase production on large scale. So we serially dilute the soil sample, heat it to kill the unwanted microbes, vegetative cells, inoculate on the surface of starch agar, you can see here in this diagram, add iodine after incubation, and enzyme producing microorganism, the colonies of enzyme producing microorganism will be surrounded by a clear zone of hydrolysis. They are picked up. You can see here in the next slide, the whole plate has turned blue because starch is present in it, and the colony is surrounded by a clear colorless zone. This is the microbe which produces enzyme amylase. Okay, so enrichment culture technique can be used for isolating amylase producing organism from soil. The fourth method or the fourth procedure for primary screening is use of indicator dye. We call it indicator dye technique. Indicator dye technique is especially used for isolating organic acid producing organisms and amine producing organisms from natural sources. Okay, so microorganisms that are capable of producing organic acids or amines from natural source can be detected by using this method, that is indicator dye. And in this method, what do we do? When we prepare any medium, when we prepare nutrient agar medium or any growth medium, we add pH indicator dyes. So pH indicator dyes are added to the medium that is used for this technique. What are the pH indicator dyes? We can add bromothymol blue, we can add neutral red, phenol red, any such indicator dye. But it should be a pH indicator dye. Okay. So microbes capable of producing organic acid or amines from natural sources can be detected and isolated by incorporating pH indicator dyes like neutral red or bromothymol blue in nutrient agar medium. Okay, then the medium is inoculated with microbes from soil. Okay, or we inoculate it with soil sample. After inoculation, the plates are incubated. After incubation, we check for the colonies that are surrounded by, okay, or the colonies which are able to produce after incubation. If the soil sample contain microorganisms which are capable of producing organic acids or amines, if, say for example, if any microorganism is able to produce organic acid, it will release acid into the medium, and as soon as acid is produced by the microorganism, the color of indicator dye will change. So we can see a color change in the vicinity of the microbial colony. So the change in the color of a particular dye in the vicinity of a colony will indicate the ability of that colony to produce any organic acid or a base that is an amine. Okay, so we take a medium containing indicator dye, inoculate with the soil sample, incubate. After incubation, check those colonies which are surrounded by different color. Okay, say for example, if we have added bromothymol blue into the medium and prepare the medium, the pH will be neutral. At that time, the color of the medium will be green. If the colony is surrounded by a yellow color, it indicates that the organism is able to produce acid, because at acidic pH, the color of bromothymol blue changes to yellow. Okay, so change in the color of the particular dye in the vicinity of the colony indicate that the organism is able to produce organic acid or amine. Okay, these colonies which are surrounded by different color are identified, isolated, and further purified by repeated subculturing or streaking on stock culture. Okay, and then they can be used after secondary screening. They can be used for large scale production of organic acids or amines. Okay, so here we finish with all the four methods of primary screening. Number one, crowded plate technique for isolation of antibiotic producers from natural resource like soil. Number two, oxanographic technique for isolating growth factor producing organism. Number three, enrichment culture technique for isolating specific kind of microorganism like enzyme producing organisms from soil. And number four, use of indicator dye to isolate organic acid or amine producing organisms from soil. Now, all those microorganisms which are isolated by primary screening are subjected to secondary screening to make sure that they really are of importance. They really are of importance to the industry. Okay, so whether they really produce the product on large scale, whether they are reliable, what are the physical, chemical properties, what are the growth requirement of the microbes that are isolated by primary screening needs to be checked, and that is all done by secondary screening. So primary screening of microorganism is always followed by secondary screening. So next time we'll discuss about